Hello, this is Dr. Clark, and today we're going to be going over the digestive system. So when we look at the digestive system, it's going to also be known as the alimentary tract, the gut, and the gastrointestinal tract. Gastrointestinal tract really means more from the stomach to the anus, gastro meaning the stomach, intestinal meaning large and small intestines. And the digestive tract actually means more all the way from the mouth all the way back to the anus. And pretty much it's a tube that runs through the animal's body, starting at the mouth where food is going to enter. It's going to be passed through the esophagus and to the stomach where acid's going to start digesting it, back to the small intestine where digestion continues and food is going to be absorbed, and into the large intestine where it's going to be partially fermented and more nutrients pulled out and it's going to be absorbed, and then finally to the rectum where feces will be stored and then it'll be passed out through the anus. So there's a lot of species variation when we start looking at the digestive tract. And so some of the things is going to be herbivores, they're going to be plant eating animals, cattle, sheep, goats. You also have horses that are in this list. And so they have some differences that are unique to their digestive tracts. You have true carnivores like cats that they eat strictly meat and nothing else. And then you have omnivores, which are like dogs, bears, and humans. And so we actually eat both plant and meat materials. Monogastrics have a simple stomach, so the stomach is just one large compartment. And a ruminant is actually an animal that is a herbivore that ends up having, they have one stomach, but that stomach ends up having 4 different compartments with it. So when we look at the digestive tract, there's 4 major functions that come along with it. First is prehension of food, so this is grasping. This is done in different ways. Primates like us, we use our hands for prehension of food. Horses use their lips. Dogs and cats use their teeth. Cattle and a lot of other ruminants actually use their tongue to grasp food with. Mastication, this is the mechanical grinding or chewing up of food, so it can be broken down into smaller bits, so chemical digestion has a more effective way of breaking down the food. Then you have chemical digestion that's going to happen. It's going to actually start with the saliva in a lot of different species. And it's going to go on into the stomach and small intestine, where in the stomach you're going to have acid and pepsinogen that's going to break down proteins and different materials in there. And then the small intestine, you'll have more pepsinogen, things like amylase, which breaks down sugars, lipase, which breaks down fats. And it's going to break them down into individual things like amino acids, simple sugars, so monosaccharides and disaccharides, and simple fats that can be easily absorbed into the body. Also, things like minerals and vitamins are also going to be absorbed, and a lot of these are broken down through chemical digestion so they can be absorbed across and into the body. And of course, then you have things such as, with that chemical digestion, you do have those absorption of nutrients we just talked about, and water being the main nutrient right there that the body needs besides oxygen. The last thing that it does is it eliminates waste, and it does this through several different ways. One is through bile that's actually put in the small intestine to mix with food and help with digestion. But there's certain things that are excreted in bile that's going to be eliminated from the body. Then you also have things that are actually excreted from the cells in the intestinal tract into the lumen to be removed from the body too. Well, look at the structure of the digestive system. You have the inner lining, which is the mucosa. In the mouth and around the rectum, this is going to be usually squamous, and it's going to be stratified, multiple layers. And it's going to be from the mouth down through the esophagus. It's going to be stratified squamous. Once you actually start getting into the stomach and the small intestine, large intestine, the cecum, this is actually going to change over in the mucosal lining to be more of a simple columnar ciliated epithelium. Below that you have submucosa, so this right here would be your mucosa. You have below that the submucosa, which is going to be more like the dense connective tissue, kind of like the dermis, and it's actually going to have blood vessels and lymphatic vessels and everything to hold the mucosa in place and to support it and get nutrients to it, and also be able to have a way through like the veins and lymphatic vessels to actually carry nutrients away from the digestive tract into the body. You have the muscular layer outside of that, which is also known as the muscularis. And then finally you have the outer serosal layer. So this is going to be that outer tough lining on the outside of the intestines. It's going to be a lot of times glossy. It's going to have uh, basically some viscera that goes around it and keeps it from helping to stick to other organs. Also, part of the cirrhosis is actually going to be formed from what we call mesentery. And so mesentery actually comes down from up around the spine and actually supports and holds all these intestinal organs in place, and also gives a way with the mesentery of actually having blood vessels come in and like arteries coming into the part of the intestinal tract, veins actually leaving and carrying blood away. And then you also have lymphatics that are going to run through this mesentery and nerves. And so that mesentery is that sheath of connective tissue that actually suspends the organs like we were talking about. And they contain all those things, the blood vessels, the lymphatic vessels, and the nerves in the GI tract. Kind of talked about this is the stratified squamous epithelium is going to be thick and tough. You're going to find it in the mouth, the pharynx, the esophagus, and the anus, and the rectal area. And then simple columnar epithelium is going to go from the stomach all the way back through the large intestine. And the simple columnar epithelium is going to be ciliated, and this is really important in being able to actually absorb a lot of your nutrients, because that ciliation really helps to improve and increase surface area for food to actually be brought in, or the nutrients to actually be absorbed and brought into the body. When we look at it, most of the digestive tract musculature is actually going to be a smooth muscle, which is involuntary, and it's going to move in an aboral direction. But there is some skeletal muscle that's going to be found in the mouth, the pharynx, and in the esophagus. Remember, smooth muscle is always going to move in one direction, so smooth muscle in the body moves aboral. But skeletal muscle, anytime that you have a muscle that moves in one direction or an agonist, you always have one that moves in opposite, an antagonist. So things like being able to regurgitate food for cows with the esophagus, it's very important that they have skeletal muscle in there, that they're able to actually regurgitate food and be able to chew their cud and swallow it. For us, it allows us to actually vomit. Some animals don't have skeletal muscle throughout their esophagus, like horses and rabbits. And one of the things that consequently happens with them is they're not actually able to vomit. One thing about the skeletal muscles in the mouth is it allows for chewing, and that chewing allows for saliva to mix with food. In a lot of species, saliva actually starts the digestive process. It also helps to lubricate the food so it can easily slide down the esophagus and make it to the stomach. And a lot of the skeletal muscle in the tongue is very important in initiating swallowing. So the smooth muscle is the majority of the esophagus. There's some skeletal muscle mixed in most species, except for the ones that I mentioned, and it's going to be found throughout the stomach, small intestine, large intestine, back into the rectum. There's actually an internal anal sphincter that is smooth muscle. The external anal sphincter, the one that you have voluntary control of, is actually skeletal muscle. So the majority of the digestive tract is actually made up of smooth muscle. And it's arranged in both the circular and longitudinal layers. And so what this does is it actually allows it to contract and pinch down, and then actually allow that pinching to actually move forward to help actually move food through what we call peristalsis. So this is actually showing peristalsis right here. So here's a food bolus. So this contraction would start right here, and the circular muscles would actually contract down, pinch it off, and they would isolate this food bolus and actually make it start moving away from where that pinching is. And then why it's in longitudinal section is then this area pinches next, and this area pinches next, so on and so forth. So what you actually get is this food bolus being moved forward. So it actually is a wave-like motion that actually propels the food. One interesting thing that happens is these are all segmental contractions, so we call them peristaltic segments or segmented areas. And so it's not like all the intestine contracts all at once in one solid wave, because if it happened, food would go right through you in no time. And so what actually happens is this area right here is contracting and pushing food this way, and then this area will contract after the wave gets here and pushes food this way. One interesting thing that happens is the food that's caught right here in between where this contraction starts, part of it is going to be squeezed. No different than like if you took something like Play-Doh and squeeze it between your fingers. Part of it's going to go, say if it's between your right thumb and right forefinger, part of it's going to go towards the right and part of it's going to go to the left. So this actually allows mixing of food. So some food will be squeezed back this way, other will be propelled forward. Some of that food that's propelled forward, once again, is going to be squeezed, part of it pushed back, part of it propelled forward. And so this actually allows for movement of contents. And then, of course, as there's relaxation of these contractions, it allows some of that food to actually spill back and forth. And so this actually allows churning and mixing. Overall movement of the food is going to be aboral, but part of it's going to be pushed back, part of it's pushed forward. And it's this whole stirring and mixing and moving around the food actually helps with chemical digestion because it actually helps to break up that food into smaller particles and stirs it up to where all the enzymes and all the chemicals can actually get to it to break down. The other thing it does is it greatly helps in absorption. And so the way that it helps in absorption is by stirring it up constantly. It's meaning that all the food's actually being able to touch that ciliated column or epithelium and get to those little cilia that are sticking out there and let them actually absorb the nutrients that are needed for the body. When we look at the buccal cavity is going to be the mouth, or the oral cavity is another name. It's going to consist of the lips, the tongues, the teeth, salivary glands, hard palate, soft palate, and oropharynx. Lips play a major role in a lot of species in actually prehending food, so like in horses. Other species, it plays a role in actually helping to keep food in, such in cattle. As in cattle, they don't really use their lips to get food, but it's very important for them to be able to keep food in. You're also going to find salivary glands in there that are going to produce saliva. There's 3 pairs. There's the parotid salivary gland, which is right below your ear, the mandibular salivary gland, which is right there at the bend of your mandible, and the sublingual salivary glands, which are actually up underneath your tongue. And depending on the species is what you're going to find in the saliva. Some species are going to have amylase in there to help break down sugars, like we have amylase in our saliva, so does dogs and horses. Some might have lipase, and this is usually found in a lot of young animals that breaks down lipids, so young animals that are nursing. Some just have saliva like cats that is just primarily used to help get food lubricated and swallowed down. Teeth are used for mastication and chewing, and so this is actually physically breaking down the food, which is really important because by breaking it down physically, it makes it to where then those chemicals of digestion can actually get in there and start breaking down the food. Or in the case of a ruminant, it actually gives that surface area needed for the bacteria to actually start breaking down the food. When we look at the teeth, we have the upper arcade. This is going to be in the incisor bone and the maxillary bones, and the lower arcades, which are going to be in the mandible. When we look at teeth shape, there's a difference between carnivores and herbivores. So the tooth over here on your right is going to be that of a carnivore tooth. They're going to be more pointed on their occlusal surface. They're going to slightly turn back towards the mouth. So this is an example of a dog tooth here. And they're used for holding on to prey and tearing and cutting and shredding on these carnivore teeth. Whereas the herbivore teeth are a lot more flat. And so they're meant more for grinding. So the one A over here, the one towards your left, is that of a horse. And so you can see it's a lot flatter. It's not as pointed. So it's actually meant for grasping and tearing off forages instead of meat. There's some other things you can see here is the fact of one thing is the root canal goes a lot further up and is a lot thicker right here in this pulp cavity in a carnivore tooth, whereas in a herbivore tooth, it's a lot more narrow when you get up here into the crown, and it doesn't travel quite as far. The other thing you're going to notice is they both have an enamel and cementum, but like in horse and rabbits and rodents, a lot of them don't actually have enamel over the grinding edge. They just end up having dentin and some cementum in that area right there. So enamel's the hardest thing in the body, and so like in a carnivore, their teeth are totally covered by enamel. However, when you get to the shape of a tooth on like a horse or a herbivore, sometimes they don't have full enamel actually covering the whole tooth. Well, look at teeth. You have incisors. Incisors are used for grasping. They're the most rostral of the teeth. Next in line are canines. In omnivores and carnivores are used for tearing and grasping onto things. Horses, like stallions, have canines. However, they're used more as fighting teeth, and so they're not really used for what their original use was, for grasping onto things. Some animals don't have true canines, like cattle. They have what we call a modified canine, but it looks more like an incisor. Premolars, these are cutting teeth, so always remember that. These are the true cutting teeth. No matter if you're looking at meat or if you're looking at a plant, what actually cuts it up ends up being your premolars. And finally, your molars all the way in the back of your mouth are very important for actually grinding. So actually making things up into smaller particle sizes. When we look at teeth terminology, we've kind of gone over this before, but we'll review it really quick. Lingual means towards the tongue, palatal means towards the hard or soft palate, labial means towards the lips, and buccal means out towards the cheek. We have what we call a dental formula. And so we're looking at the number of teeth in the upper and lower arcade. And so the upper arcade is going to be the incisor and the maxilla. The lower arcade is going to be the mandible. And we actually split the mouth into quadrants. You have the right upper quadrant, you have the right lower quadrant, the left upper quadrant, and the left lower quadrant. And the way that we end up splitting these up is from the right middle incisor all the way back around to the premolar would be 1/4 of the arcade, and that would be half of the upper arcade. And then from the middle incisor back over to the back of the molar on the right lower would be 1/4 quadrant also, or 1/2 of the lower arcade. We have some designations for the different teeth when we're looking at the dental formula. I is for incisor, C is for canine, P is for premolar, and M is for molar. If they're uppercase, that means they're adult teeth. If they're lowercase, that means they're deciduous. We have something we call the rules of 4 and 9s. So canines are always numbered, when we get to the Trident numbering system, they're always numbered number 4, and that's because they're the fourth tooth. There's usually 3 incisors. The fourth tooth you get to is going to be a canine. And then the 9th tooth in line that you're usually going to get to is going to be a molar. And so using the rules of 4s and 9s, this actually helps us out when we teach you the Trident numbering system. But one of the things that's important to know is when you're sitting here and actually looking at the teeth, premolars, canines, and incisors all have deciduous teeth. So we can have a lowercase I, lowercase c, and a lowercase p. However, molars are only permanent teeth, so a molar should only be capitalized. You'll never find a molar that's going to be a deciduous tooth. Ruminants don't have any upper incisors or modified canine teeth. They have a dental pad in its place. So when you look at a ruminant's dental formula, they're never going to actually have incisors or canines on their upper up in their incisional bone or incisor bone area. They're always going to just have them down in the mandible. So we have the tooth type followed by a number / by a mark. So they're going to kind of show it here, like the first incisor is going to be, like I1, I2, I3. And so we can actually count these off. And so the first number, when we're looking at the dental formulas, of course, C would be for canine, and then we have P for premolar, so that can be premolar 1, 2, 3, and 4, and then molar 1, 2, and 3. We actually have a way that we can actually sit here and actually look at the teeth and actually count them out by looking at how many are in each quadrant of the arcade. So, and when we do this, we're looking at something we'll see on the next chart, and it'll make sense when they're talking about half the number in the upper and half the number in the lower, and then multiplying by 2. So this is what they're talking about right here, is lowercase I. So if we took the right upper quadrant of the mouth, there's going to be 3 incisors there, and this slash right here means the lower quadrant in the mandible, so there's 3 incisors. There's 1 canine right up here in the maxilla. There's 1 in the mandible. There's 3 premolars in the maxilla. There's 3 premolars in the mandible. And that's for a puppy. We add those together, so 3 plus 3 is 6, plus 1 plus 1 is 8, plus 3 plus 3 gives you 14. So there's 28 teeth in a puppy's mouth. When we go to the adult, you're going to see that we have 3 incisors in the right upper quadrant or left upper quadrant, 3 incisors in the lower quadrant, so 3 in the incisor bone, 3 in the mandible. We have 1 canine in the maxilla, 1 canine in the mandible, 4 premolars in the maxilla, 4 premolars in the mandible, 2 molars in the maxilla, and 3 molars in the mandible. And when you add all these up, these numbers up here, so 3 plus 3 is 6, plus 1 is 7, plus 1 is 8, plus 4 is 12, plus 4 is 16, plus 2 is 18, plus 3 is 21. So we take 21, we multiply by 2, we get 42 teeth in a dog's mouth. Cats are a little bit different. They're actually missing a few premolars. They have only 1 molar each. They have 3 premolars on the top, 2 molars on the bottom. They have 30 teeth total. Horses get really fun and confusing because it depends on if we're talking about male or females. Male horses have canines, females don't. So a female horse automatically is missing the canines. So you can -4. One thing is some horses have something called a wolf tooth, which is their first premolar, but others do not. So that's the reason why you see 3 to 4 right here, just depending on the horse. So horses can have anywhere from 36 to 42, depending on if they're a mare and a stallion. If they have a stallion with canine and all of his upper premolars is going to have 42 teeth. A mare that does not have any, she won't have any canines, and she says it only has 3 premolars. She never got a wolf tooth, so she doesn't have that fourth. She'll have 36. Kind of the numbers you're looking at is, if they ask you, is going to be for a mare somewhere between 36 and 38 is the number of teeth they should have, and for a stallion 40 to 42. If they ever ask you on the national boards, generally for horses, if they give you 2 answers within that range, you just have to guess the best. But if they tell you specifically for a mare, they're usually looking for 36. If they're looking at a stallion, then it's usually 48. You might add 2 onto that and make it 38 and 42. But that's what they're going to ask you. So horses get a little bit fun and confusing with that. And we'll go over this again later in equine. Pigs, porcine are the only ones that have a full complement of teeth. They have 3 incisors top and bottom, 1 pair of canines top and bottom, 4 premolars top and bottom, and 3 molars top and bottom for a total of 44 teeth. So they're the only domestic animal that has a full set. Bovine, they don't have any incisors or modified canines on the top. They just have 3 incisors in the mandible and 1 modified canine in the mandible, but they still have 3 premolars and 3 molars, just something similar like a horse, for a total of 32 teeth. So this is the dental formula for domestic animals, something that you need to know, not only for in here, but also in practice. It's important to know when you're doing dentals how many teeth are supposed to be in the mouth. If you're doing dental work on a cow or a horse or a dog or a cat. Also, this is a question they love to ask on the national boards. When you look at the structure of the tooth, you have the outer part of the crown, which is going to be capped with enamel. Inside of that's going to be dentin. So one thing about enamel, it's the hardest substance in the body. The other thing is, as you actually age, enamel actually lose blood flow, and actually, it's not really dead tissue, but it doesn't actually replace itself. So it puts itself on in the younger animal, and as you get older and it wears off, it's not actually replaced. Inside of that is dentin, which is kind of like living tissue. It's kind of like a really dense cortical bone. Adds some strength and structure to your enamel below and gives strength and structure to the tooth. Inside of that, you have the pulp cavity, which is actually rich in nerves and blood flow to actually keep the dentin alive. You also have something that's called cementum along the root here underneath, and it's more like kind of like a compact bone, not as dense as enamel is, cementum. And it actually has a lot of ligaments attached to it that actually help to keep the tooth rooted to the bone in the socket. And then you actually have the gingiva out here, which is the gums, which actually is soft tissue that actually helps to protect anything from getting between the cementum of the tooth and the bone that it's actually attached to. And we kind of already talked about the enamel out here, and then this epithelial layer of the gingiva that actually helps to protect. Gingiva is fairly forgiving, and so it can actually take trauma and actually heals fairly quick. When we look at the oral cavity, it's there to prehend food, to actually chew it up and masticate it. And then you can actually add saliva, which can start indigestion. And so a lot of omnivores and herbivores actually have things like amylase within saliva that actually helps to start to break down any simple sugars within the food. Animals like cats don't actually have amylase, so your carnivores, they're not actually present in. There's no amylase actually within their saliva. We actually go to looking at saliva in cattle. There's actually a lot of bicarbonate and urea that they actually add to actually aid in rumination and the bacteria within the rumen, trying to keep it healthy at the proper pH. And actually the urea within their saliva also helps that bacteria to start making proteins and aids in its digestion. And then, it's very wet, saliva is, so it actually helps to prepare the food for swallowing. Those digestive enzymes we talked about, amylase is in saliva of omnivores. You're also going to find it down in the pancreas, and it's actually there to actually start breaking down starches or sugar. Lipase we usually find in young animals that are on a milk diet that actually starts breaking down fat. And then we talked about the bicarbonate found in the saliva of cattle, which actually helps to kind of neutralize any acidity within the rumen. The digestive system is controlled through the autonomic nervous system. The main nerve that's going to feed into it is going to be the vagus nerve. You have parasympathetic stimulation, which increases salivation and actually increases GI motility. And then sympathetic stimulation actually stops secretions like saliva and actually inhibits GI motility. Sometimes we actually have a problem with this, like with some of our anesthetic agents, which actually increases some of our parasympathetic stimulation. It takes away sympathetic, and so therefore we get things like vomiting, or we'll get excess salivation. And so we actually use a parasympathetic blocker like atropine or glycopyrrolate, what are known as anticholinergics. And what they actually do is they don't really boost the sympathetic nervous system. They block the parasympathetic, and they stop salivation and things like vomiting from occurring. Anything that increases the sympathetic nervous system, like giving epinephrine or something like that to an animal, is actually going to sit there and actually stop the GI tract from moving. It's actually going to stop saliva production. The esophagus is the tube that transports material from the pharynx to the stomach. It enters the stomach at an angle in the region of the stomach called the cardia. There's actually a sphincter right there that actually tries to control and make sure what's in the stomach stays in the stomach, that the acid in the stomach doesn't actually travel its way back up in the esophagus where it can actually cause problems. The stomach actually can expand and fold against the esophagus and actually kind of keeps the lower end of the esophagus closed, which actually helps to keep from reflux happening. Sometimes this is a really strong muscle, like in horses and rabbits. Also, horses and rabbits aren't really able to have reflux or vomit because they also lack that skeletal muscle within their esophagus. Monogastric, so these are animals with simple stomachs. They have 5 different areas. When we look at a simple stomach animal or monogastric, you have the cardia, which is right where the esophagus comes in, and it's called the cardia because it's right next to the heart. If you get any type of indigestion in this area, the reason why we call it a heartburn is it makes it feel like your heart's burning because it's right here in this area of the cardia. Next, you have the fundus, which is this large blind pouch right here that can easily expand out and fill with food. And there's certain glands in there that actually help to make hydrochloric acid and different digestive enzymes. Body can expand also, and as it fills up with food, and one of its main roles is also making different things like hydrochloric acid and digestive enzymes. Next, you have the pyloric antrum. This area can fill up with food too. And it's responsible for making something called gastrin. And gastrin has a role in emptying of the stomach and also plays a role in making buffers to protect the body against the stomach acid and the digestive enzyme, and actually increasing or decreasing the amount of hydrochloric acid and digestive enzyme that's in there. And finally, you get to the pylorus, and it's the sphincter between the duodenum, which is part of the small intestine, and the stomach. And its main job is to make sure that nothing refluxes from the small intestine back into the stomach. And it also controls what's going to leave the stomach, make sure that all this acid and everything doesn't leave the stomach too fast and enter the duodenum. So the cardia is that opening from the esophagus. Fundus is a distendable blind pouch that expands as food is swallowed. The body can also distend somewhat in the middle. Both the fundus and the body contain numerous glands, including the parietal glands, and these parietal glands make hydrochloric acid. The chief cells, these actually make an enzyme called pepsinogen. Pepsinogen turns into pepsin, which actually starts to break down amino acids and proteins. And then you have mucus cells, these protective goblet cells that line the stomach that actually produce mucus that has extra bicarbonate in it to actually help buffer against this hydrochloric acid that without this mucus right here, would basically the stomach would start to digest itself. The pyloric antrum, they say it grinds up food. It really doesn't. It just kind of keeps it stirred and moving around. And one thing is it actually has G cells in there that makes gastrins. And so this gastrin is a hormone that's actually secreted in the bloodstream, and it plays several different roles that we'll talk about. Pylorus is that muscular sphincter that's actually going to control movement of food from the stomach into the duodenum and actually helps to prevent backflow of duodenal contents into the stomach. When we look at gastric motility, each area of the stomach has some different motor functions. So the fundus in the body relax with swallowing of food. The body of the stomach also contracts to help to mix food, so it actually is what's important with stirring. The body is a lot of times also what's rumbling when your stomach is what we call quote unquote empty, when it's wanting food. You'll actually notice it actually stirring and moving around. And the pylorus antrum actually increases contractions in response to swallowing. So as you swallow food, its contractions actually start to speed up, and it actually helps in mixing up food and actually moving that food through the pylorus over into the duodenum. Peristalsis starts in the stomach and is going to continue down in the intestines. So that movement of the digestive tract starts in the stomach, and those signals are going to be continued down through the intestines. The stomach has the mucosal layer, the submucosal, and the muscular layer, and also an outer serosal layer. There's smooth muscle that responds to hormones, peptides, and other nervous control like the vagus nerve. And then you have parasympathetic stimulation actually causes the fundus to relax. It increases the contractions in the pyloric antrum. Sympathetic stimulation actually stops all motility and stops the stomach from moving whatsoever. Gastrin produced by G cells in the antrum of the stomach, and that pyloric antrum, they do two things. First of all, they increase production of hydrochloric acid to help digest food. And then they actually stop some of the muscle activity of the fundus. So they actually have an inhibiting effect on it, trying to give a chance for the food to be digested more by the hydrochloric acid up in the body and up in the fundus. The enteric or enterogastric reflex is distension of the small intestine, increases acidity in the duodenum, which stops stomach contraction. So anytime that we get too much acid in the duodenum actually tells the stomach, and specifically the pyloric antrum, to stop sending food its way until it can get that acid under control. If it doesn't, then we actually get what they call duodenal ulcers. So the main thing it does is it actually slows down this enterogastric reflex from the small intestines. Actually, when they get distended or they get too much acid in the duodenum, actually delays gastric emptying. Secretin released from the duodenum in response to excessive stomach acid actually causes the fundus to relax more so the stomach can hold a lot more food, and actually can stop peristalsis of both the body and the pyloric antrum of the stomach to slow down gastric emptying. The other thing is something called CCK, or cholecystokinin, and this is released in response to large amounts of fats or proteins in the duodenum, and these also slow down contraction of the stomach in the pyloric antrum, body, and fundus. This is something that happens like after Thanksgiving. You eat a lot of turkey or a lot of fat foods, and you get that really full feeling, and you feel like you can't eat any more. That's because everything's slowed down in your stomach. It feels full and distended because of this CCK. All that extra protein and fat's gone to the duodenum, and the duodenum has said, slow down, give us some time to digest this, don't send anything else our way. Secretions, the pepsinogen by the chief cells, they're precursors to pepsin, and pepsin is very important in breaking proteins back down into amino acids. There's also something called intrinsic factors, which are actually secreted by certain glands in the submucosa. And these intrinsic factors are very important for absorption of B12 into the body and through the small intestine. So a lot of times B12 is broken down in the stomach, or its components around it are, and then intrinsic factor actually attaches to that B12 and allows it to actually be transported to the small intestine where it's absorbed. The mucus in the stomach is actually a fairly complex substance, and it actually provides a coating against the things like the pepsin and pepsinogen and against the hydrochloric acid, and is produced by specialized goblet cells. And most of it is mucus in there, but also this mucus actually contains a lot of bicarbonate. And as we learned at acid-base balance, bicarbonate is a very important buffer, especially against acids. And so what it actually does, the bicarbonate does, is it tries to control the amount of acid in the stomach and make sure that it doesn't overwhelm the mucus and start digesting the mucus itself. Hydrochloric acid is actually going to be made through little pumps, proton pumps in those parietal cells. And all it is the body takes hydrogen ions and chloride ions and secretes them together in these parietal cells and makes hydrochloric acid. And they actually go out, and that hydrochloric acid starts breaking food apart and makes it easier to chemically digest. A lot of times these parietal cells are going to be influenced by things like gastrin. So gastrin plays a role, and hydrochloric acid increases the amount. There's certain forms of histamine that actually sit there and increase the amount of hydrochloric acid made. And acetylcholine also plays a role in making these. And a lot of our different drugs that you know, like Zantac or ranitidine, they work on something either like gastrin or histamine or acetylcholine, and they actually stop that production of hydrochloric acid. Prostaglandins are very important. So when we look at prostaglandins, they're very important at inhibiting gastrin release, therefore slowing down the amount of hydrochloric acid being made. And then they actually stimulate bicarbonate and mucus production in the stomach. They also enhance blood flow to the stomach, and they actually are important in making sure that macrophages and mast cells are very active in the stomach, and mast cells are basophils as far as immune production. They're also very important for stabilizing the gastric cells themselves against the hydrochloric acid. One thing is when we look at these prostaglandins is we have to think about NSAIDs. A lot of our NSAIDs are not selective enough against prostaglandins for inflammation, and those prostaglandins that are needed to produce stomach acid and to protect the stomach. And so long-term use of some of these things, such as ibuprofen in people or phenylbutazone in horses, or even something like Rimadyl in dogs, can actually lead to gastric ulcers because they're blocking these healthy prostaglandins. And so we've gone a lot more in medicine towards what we call selective. There's no NSAID out there that does not have no effects on the stomach prostaglandins, or what's known as cyclooxygenase 1 or COX-1. But we do have ones that select a lot less for that and a lot more for cyclooxygenase 2 or COX-2, which is a prostaglandin. And we like to go with those. And so those are going to be things like meloxicam, Previcox, Equiox, a lot of those new drugs on the market that are much more selective for inflammation and a lot less selective for these that actually protect the stomach. So they greatly reduce the chances of having some type of issue with the stomach, such as ulceration. Ruminants, when we look at it, there's one stomach with 4 compartments. So I know here it says one true stomach and 4 stomachs, but you know, it's one stomach with 4 compartments. And so what ruminants do is they actually pretty much try to shovel in all their food whole, and they let it start fermenting in their stomach. And a lot of times, a lot of ruminants will sit there. They eat an average of 12 hours a day. They ruminate an average of 10 hours a day. So they try to get all the food they can in as fast as they can. They chew it a little bit, they swallow it, and then they go, and after they get kind of their rumen full, they go lay down or they stand someplace and they start to ruminate. And so what they actually do is they actually take some of this food and they regurgitate it back up in a bolus and they chew it and then they re-swallow it. And by them chewing it and re-swallowing it, it actually helps to break down that fiber better for the bacteria, but that extra saliva actually helps to also control the pH within the rumen. When we look at it, the abomasum, they call it the true stomach. I prefer to call it the glandular stomach. So this is the part of the stomach that would be like the body, and it has the pylorus and actually a pyloric antrum. And this is where your gastrin cells are, your parietal cells and your chief cells are all in the abomasum. When we start looking at the other stomachs like the reticulum, the rumen, and the omasum, the reticulum is the smallest part. It's the most cranial part of the compartments, and so it would be right here. It's where food actually comes into initially. It's forced up from there into the rumen, and then it also plays a very important role with its honeycomb shape of being able to actually make that bolus of food to be regurgitated and rechewed by the ruminant. When we get to the rumen itself, it's a series of muscular sacs that are separated from one another by these folds or these pillars, and it's very important, and it has this regular contraction. Every 2 to 3 times a minute, there's a rumen contraction that goes through, and this rumen contraction ends up being very important in stirring those rumen contents and making it to where they can be broken down and digested by the bacteria. And the rumen and the reticulum actually have these contractions that actually go together. The other thing they do is when you have something fermented, you build up a lot of gases. So the rumen and the reticulum work together to actually allow for a ructation. And so a ructation is actually the burping or belching of excess carbon dioxide and methane gas that's actually made from rumination, from these bacteria fermenting and doing the digestion. If these are not able to be removed, then the ruminant actually can bloat, and the rumen gets very large from all this gas buildup, and it can actually kill the animal by suffocating them. Rumen motility is controlled by parasympathetic nervous system. So the parasympathetic nervous system, when it's in action, rumen motility carries on. If there's sympathetic stimulation whatsoever, then you get rumen atony. So it stops moving. The rate, strength, and contraction of the rumen is going to be determined by the pH. So there's this range of pH. It should be between about 6 and 6.5. That's very important for the rumen to actually do its normal job. Things like volatile fatty acids, which are breakdowns of carbohydrates, play a very important role in the rate and strength of the rumen contraction. Stretch receptors, so the fuller it gets, the more it's going to contract. The more empty the rumen is, the less it's going to contract. And there's actually some feedback to the brain also with the rumen from some of these stretch receptors, pH and all this, and that's actually going to give you some stimulation of the vagus nerve. So all ruminants do what we call foregut or forestomach fermentation, so they actually ferment food before it actually goes through the rest of the digestive tract. And this fermentation is going to do a couple of things. It's going to break down a lot of your complex celluloses. These complex celluloses are actually forms of carbohydrates, and they're carbohydrates that are not easily broken down by our digestive system. So we actually have a hard time of breaking down cellulose. But bacteria actually have a very easy job, and they can break down the cellulose into its independent carbohydrates. The other thing it does is these bacteria can actually break down protein and actually rebuild amino acids and other proteins from the ones that they actually break down during fermentation. Carbohydrates are going to be broken down into mostly what we call volatile fatty acids. These volatile fatty acids are different than glucose, but they can actually be run through the Krebs cycle. And so when we look at ruminants, they have a lot more volatile fatty acids and a lot less glucose in their bloodstream. And then a lot of these peptides, some will be converted to ammonia, and then they'll actually be used by the microbes to make other amino acids that can be absorbed by the animal. Volatile fatty acids, some can be converted to glucose. A lot of them are actually converted to fat tissue, milk fat, and then other components. The omasum, so from when the food particle gets to the right size and it's been broken down enough by rumination and by fermentation, it's actually able to pass out of the reticulum into the omasum. And the omasum has a lot of different folds in it. They are muscular. It used to be thought that they ground and broke down food particles. It doesn't. And it's still in the book. It's still on this slide, but let me tell you it doesn't. The main thing that it does is it has a lot of what they call mini plies in there. There's a lot of these plies. There's muscles with it. There's some contractions to it, and these contractions help to stir things through so that a lot of your volatile fatty acids are absorbed through the omasum. Also, cattle produce something like 50 gallons of saliva a day, which is a lot more saliva than they have blood. So the omasum is very important in actually bringing that bicarbonate back into the bloodstream that they're going to use to put back into the saliva, and bringing that extra fluid from that saliva back into the bloodstream to where it can be remade as fluid back into the saliva again. So it's very important in not only bringing in volatile fatty acids, some different B vitamins, but it's also very important in absorbing a lot of your water and bicarbonate from the saliva that's in the rumen back into the body for it to be reused. And then finally, said the abomasum. This is going to be very similar to the monogastric stomach. This is where you're going to actually have things like your chief cells, your parietal cells, your pylorus and pyloric antrum with your gastrin that's going to control how fast is this rumen emptying going to take place, and how fast does food actually leave the abomasum and enter into the duodenum. Newborn ruminants, one thing to know about their digestive tract is it functions very similarly to that of a monogastrics. Somewhere around 4 to 8 weeks, they actually start eating solid food and their rumen actually becomes active. But while they're on milk, and especially in their early life, the rumen and reticulum are both non-functioning. There's no bacteria in there for them to actually digest anything. And so the rate of rumen development and reticulum development is actually determined by when they actually start eating their first dry food. And we have something called the reticular groove, or more commonly known as the esophageal groove. And so this is actually the wall of the reticulum. When the calf puts its nose up into the nursing position, the esophagus actually tugs on the reticulum, and the reticulum actually makes a fold or makes a tube for milk to go directly from the esophagus through the reticulum, bypass the rumen, and directly down into the omasum and into the abomasum. And this is very important for this to happen, because if any milk happens to get over in the rumen is going to try to ferment it, and this fermented milk is going to lead to bloat, can lead to infection, and ultimately the death in a lot of calves. So it's very important that if we have to bottle feed a calf, that we actually keep their nose up in a nursing position like they would with mom, which is usually about I would say a 30 to 60 degree angle, usually about 45. And by having it up in that angle, we actually close this the reticulum and make this esophageal groove to where milk does not go into the rumen. Now, when they eat, they're going to eat with their head down, and if their head is down, then this means that whatever they ingest goes to the rumen, and this is how they actually, by ingesting different things like grains or grasses or hay, they actually slowly pick up the bacteria in the environment and the food that they're ingesting that's needed to actually get the flora to actually start breaking it down and ruminating it. Once again, the problem though is if you're trying to, like a lot of people want to be with nursing calves, they try to make it easier on themselves instead of better for the calf, and they try to get them to drink out of, like, say, something like a bucket. And by having their head down, when they ingest that milk, it's going to go in the rumen, it's going to ferment, and it's going to cause a lot of problems for them. So it's all right for them to eat with their head down, so that way it makes it into, when they're ready to eat, it makes it into the rumen. It can get the rumen kick-started and get it to develop. But when they're nursing, they need to be head up in that nursing position to where that milk bypasses the rumen and goes straight into the abomasum. Small intestine is made up of 3 portions. You're going to have the duodenum, or if you're a Yankee, it will be called the duodenum. It's the first segment. It comes from the stomach, and it's somewhat short compared to the jejunum, which is definitely the longest portion. And but this is the area where food's going to leave the stomach, come into the duodenum first. Bile is going to be introduced from the liver, and then digestive enzymes from the pancreas. Once again, things like pepsinogen, amylase, lipases, all those will come from the pancreas and enter into the duodenum. And so this is where digestion of the food in the small intestine is going to start. And one thing about it is a lot of your bile salts and your bile is actually very alkaline. So it's actually going to change food from the stomach from being very acidic to actually kind of making it more neutral or slightly alkaline. Jejunum is the longest portion. This is where the vast majority of absorption takes place of food that is ingested. And the jejunum is, probably about 90% of the small intestine, 80 to 90%. And then finally we get to the caudal section of it, which is the ileum. And the ileum is the short section, and pretty much it's just a transition from between the jejunum over to the large intestine, over to the cecum, and or the large colon. And so it actually is just this one little section right there, and it's fairly rigid, and it actually helps to regulate movement of material from the small intestine into the large intestine. When we look at it, there's that intermucosal layer, the submucosa, the muscularis, and the outer serosal once again. When we look at thickness, there's layers of thickness that are going to vary with each segment of the intestines. Usually the duodenum and the ileum are a bit thicker than, say, the jejunum is. Parasympathetic stimulation is very important. So the parasympathetic maintains normal blood flow to the small intestine, causes motility and secretions. Sympathetic nervous system stops blood flow, and actually pretty much it says little effect on motility, but if you stop the blood flow, you stop the motility. When we actually look at the intestinal mucosa, what you'll see right here is what we call a villus and a crypt. So crypt is down here at the bottom, up here is the villus, and along this is actually going to be ciliated columnar epithelium. And you have all this microvilli out here on this epithelial surface, which greatly increases surface area. And there will be thousands upon thousands of microvilli on one of these epithelial cells. And so it turns something that if you were to look at it flat, that might be, say, the size of a 12 by 12 room, actually these microvilli could give something that surface area, the same surface area as, say, half a football field, or maybe even as large as a football field. So greatly increases surface area, which greatly increases the ability for nutrient absorption. New cells are actually started right down here at the crypt. So the crypt is where all your stem cells start at, or your germ cells. And as they divide, new cells actually start pushing up against the older cells up here at the very tip of the villus. And so they actually work their way up from the crypt up here to the tip of the villus. And then eventually they're shedded off, usually by some type of traumatic force, like food coming by, sometimes by age. A lot of times when these cells die and fall off, they're actually digested and recycled by the body. These microvilli are also really important because they actually contain certain digestive enzymes, and enzymes that actually help to bring certain nutrients across into the cell. And they're actually going to cross, these nutrients are actually going to come into the cell, into these columnar epitheliums, and then they're actually going to be secreted out or diffuse out into the submucosa below, where they're going to be picked up by the venous blood, which is then going to return through the portal system to the liver. Or in the case of certain fats and proteins that aren't actually able to penetrate and come through the capillaries and go back through venous return back to the liver, they're actually going to enter the lymphatic system, and they're actually going to be turned into that fat substance, that chyme that we talked about in the lymphatics, that's actually going to move back up to the heart and be put into circulation, where it'll make its way back to the liver and be processed. So crypts are those invaginations around each one of the villus. They produce the cells that push up from the bottom, replacing the older ones on top. There's also a lot of goblet cells that are going to be in here too. They're going to produce mucus to help protect intestinal mucosa, because once again, a lot of these digestive enzymes can be dangerous to the cells themselves. And so you need some of that mucus to actually kind of help protect them You don't need so much that you block or interfere with nutrient absorption, but yet you need the right amount at the same time to make sure that these digestive enzymes are not going to actually harm the digestive system itself. Peristalsis is very coordinated within the small intestine. It's stimulated by dilation of the segment of the small intestine. And there's certain things that will stimulate it, peristalsis of the small intestine, like CCK, and those different prostaglandins that we talked about. They actually help to stimulate movement through the small intestine. The segmented contractions we talked about earlier in peristalsis actually helps to move food through the small intestine, but it also helps in mixing by what we showed, mixing back and forth the materials. And by mixing it and kind of stirring it around as it's moving it forward, it actually helps to make sure that it gets good contact with the surface of those macrovilli, so all those nutrients that the body needs can be absorbed. Digestion in the small intestine, things that are going to be brought in are going to be like electrolytes, water, vitamins are all brought across, carbohydrates, fats, proteins are chemically digested and brought across. And they're going to be brought across from the lumen of the intestine, and with the assistance of enzymes in the microvilli, they're actually going to be brought into the cell, and then they're going to be brought into the body to where they can be then distributed throughout. Carbohydrate digestion, so starches are usually converted in the lumen from polysaccharides into disaccharides. And then this usually starts in the duodenum by the pancreatic amylase, and it actually starts with the salivary amylase too. Disaccharides are actually further digested by enzymes in the microvilli that are actually going to turn them into monosaccharides to make them very easily transported against the cellular membrane of those epithelial cells. Protein digestion, that gastric pepsin, starts to break apart some of the protein into smaller chains, into smaller amino acid chains. And then there's actually 5 different pancreatic proteinases. So there's trypsin, chymotrypsin, elastase, aminopeptase, and carboxypeptidase. And these actually take what the pepsin's already done in breaking down protein into smaller amino acid chains and breaks them down into their individual amino acids to where they can then be easily absorbed across the intestines. And then there's actually certain peptidases within those microvilli that actually help to actually bring across those amino acids, some dipeptides and some tripeptide chains, but mostly the single amino acids. Fats are agitated in the pyloric antrum. The stomach acid actually helps to break them into fat globulates. And then bile acids from the liver are actually going to coat these fat droplets in the duodenum, and it actually allows these pancreatic lipases to actually then, the bile salts, to actually penetrate the bile coating and actually digest those fat molecules of glycerol into smaller fatty acids and into smaller micelles that can actually then be absorbed by the microvilli into the body. Large intestine, you're going to have the cecum, the colon, and the rectum. There's variation in structures in animals. Carnivores have a very poorly developed cecum and a very short colon. Omnivores have a longer colon. Some have a more developed cecum than others. We have a very poorly developed cecum called an appendix. Dogs also have a very poorly developed cecum. Something like a bear that's an omnivore actually has a little bit more developed cecum because they eat a little bit more plant material than we do. And their primary functions can be recovering things like fluid and electrolytes, so drying out feces and storing feces until they can be eliminated. But when we look at things like hindgut fermenters like horses and rabbits, the large intestine plays a very important role in fermentation and actually making things like volatile fatty acids. A lot of your different B vitamins can be made within your large intestine and be absorbed right there. And then they actually have a lot of different proteins that are actually made through fermentation that's going to be absorbed into the body in those areas. And they have these very large, complex large intestines with large, huge cecums and very developed colons that are aid in actually fermentation and absorption of those nutrients. So this is actually showing a horse's. So when you look at a horse right here, carnivores are very poorly made, but non-ruminant herbivores like horses, and a horse right here, this is the cecum, the very tip of it's known as the appendix or the apex. This actually runs pretty much down from the horse's right hip, just about up there to its diaphragm on the right side. So it's actually quite large. And then you have the right ventral colon, which runs down the right side of the horse till you get to the sternum. It turns at the sternal flexure. From the sternal flexure, it's going to go down here in the left ventral colon. All of these actually have these little marks that you're seeing on here called haustra. And these haustra, these little compartments, are very important in peristalsis for mixing and stirring what's being fermented, and actually aids in fermentation and stirring. The other thing it does is it actually helps to also aid in absorption of things like protein and volatile fatty acids. Coming back here to the left pelvic bone, you have the pelvic flexure, and it says pyloric flexure. That is not correct. That should say pelvic flexure. And the food's actually going to move up here, and then it's going to go into the left dorsal colon. The dorsal colon does not have nearly as much haustra on it, and it's more in line for absorption of nutrients. And food moves a little bit faster through the dorsal colon. It's going to move all the way down the left side till it comes to the diaphragm, where you have the diaphragmatic flexure. And then from right there, it's going to the right dorsal colon. Here they show the right dorsal colon, and they do this in the books. A lot larger than what it actually is. It's a little bit smaller in size. It's not any bigger or wider than what the right ventral colon is. The right dorsal isn't. So it's a little bit smaller than this. But it's actually going to then move up, and it moves down the right side to where we get in horses something called a small colon. You don't see this in other species, but in the small colon is actually where a lot of nutrient absorption and specifically water absorption takes place. So in the small colon, it's very important with it that it actually takes and reabsorbs a lot of the moisture from the food, and actually what's what causes horses to have that manure and what we call that horse apple, or that oval manure shape that's fairly firm. And part of this comes from the fact that horses actually developed in an arid and a desert area. So they kind of come from North Africa along the Arabian Peninsula, our modern horses, and all the way up into what they call the steppes of Asia, which is like where the Gobi Desert is. So they actually come from our modern horse from a very dry area. So they do a lot of water conservation. So our hindgut digesters are going to be things like horses, guinea pigs, rats, and rabbits. So they've modified their large intestine to be able to ferment, produce those volatile fatty acids, produce those proteins, and bring them in just like ruminants do. And they actually do have different amounts of bicarbonate and mucus that are going to be actually produced by their colon and their cecum, and as a way to actually try to keep pH at the proper level and try to buffer the pH within the large intestine. And once again, their large intestine pH is not that different from what you're wanting to see within, say, a ruminant. Ideally is pH between 6 to 6.5. The rectum is the terminal portion of the large intestine where feces are stored. And it's underneath control of the nervous system. There's two different sphincters there, one that's controlled by the vagus nerve, that smooth muscle, one that's got external sphincter that has control there at the anus, that's controlled by skeletal muscle that actually gives you some control over when and where you defecate. If the internal gets overridden, sometimes it can be very easy for the external to get overridden too. So just remember that. It's important when people do house training. There's only so much that the external sphincter can actually hold. So sometimes people, when they're trying to house spray puppies, they don't understand why they defecate in the house. Well, they can only hold it for maybe 2 or 3 hours, and then they need to go outside. The rectum does have some lubricating glands in it that actually helps in the passage of contents. And you actually have stretch receptors in here that actually stimulate that defecation response. The anus has that internal and external sphincter. And as the rectum distends, those stretch receptors in the wall actually cause relaxation of the internal sphincter. And that in turn signals for the external sphincter for the urge to defecate. And then that will relax and allow for defecation to take place. There's anal mucosa receptors that sense the need for that defecation. You're also going to find things like anal glands and modified anal glands within this area. Other things that we come across in the digestive system is going to be things like the liver, which is very important for producing bile. And in bile, you're going to have bile acids, cholesterols, and bilirubin. And the liver is full of these bile ducts that actually take these basically waste products from the body, and they actually deliver them down in the species that have a gallbladder to the gallbladder for storage. And release of this bile is going to be stimulated by CCK. So in the response to CCK, that hormone from the duodenum, the gallbladder will contract and actually release its contents. One thing to know is there's some species that don't actually have a gallbladder. So rats, horses, and deer actually lack a gallbladder, and they just secrete bile into their digestive tract constantly. Of course, all those species like to eat constantly too. So the bile is very important for taking toxins out of the body, for flushing out infectious agents and so forth that come in the GI tract, and also is very important in actually helping in digestion and actually breaking down certain nutrients and helping them be absorbed from the digestive tract. Pancreas has a function in the endocrine system for making things like insulin, but it's also very important in making those digestive enzymes of things like we talked about, the amylase, the proteinases, and the lipases. And so these are all secreted into the duodenum to actually help break down food into its individual nutrients to be absorbed by those microvilli. It also produces bicarbonate, which actually helps to protect the duodenum from acid that's coming out of the stomach. And I said it makes the insulin and the glucagon also. A couple of things to know about the digestive system real quick that we talk about is pain in the digestive system. And so what's important to know about it is, and something they don't cover in the book, is that pain in the digestive system is related to one or two things. Either ischemia, lack of blood flow, will actually cause pain in the digestive system. The other thing is stretch receptors. So what's amazing about the digestive tract is that you can puncture the digestive tract and you will not feel pain. But if you stretch the digestive tract, it causes immense pain. So, it's one of the comparisons we like to give you is, I could lay your intestines out in front of you and take a Ginsu knife and chop them up, and you would never feel a thing. However, if I were to take your intestines out in front of you and stretch and pull on them, it causes immense pain. And so that's something very important to understand about the GI tract, that when we see pain with it a lot of times, it comes from the fact of stretching from things like bloating or distension. The other thing that will cause it, once again, is ischemia, lack of blood flow. So if we have anything that creates a lack of blood flow or creates distension, that's what actually causes pain in gastrointestinal organs. That's been the gastrointestinal tract.